Welcome to the lecture on enzymes as catalysts. There's actually two lectures in this series. The first one will talk about enzymes and their basic properties, and the next lecture will talk about enzyme kinetics and inhibition. As always, here's the lecture objectives, and you'll see them. Remember, it's a great place to start when you're doing your study, and we'll go through these in the lecture. So enzymes are proteins, but they're special proteins. They have an arrangement of amino acids in an area called an active site that allows them to act as catalyst and to catalyze chemical reactions. And if you recall the term catalysts, a catalyst is a substance that can change the rate of a chemical reaction. Usually catalysts will speed that reaction up. That's typically how we find this to be the case. And so enzymes are biological catalysts that help carry out biochemical reactions. Now, what's going to be very important as we go through this is the catalyst will increase the rate of a reaction, but it doesn't change the equilibrium constant for the reaction. It doesn't change the free energy of the reaction. It just simply makes the rate go faster. So the problem we have in our cells is most of the reactions we need for life, most biochemical reactions will not proceed at a rate that's necessary for us to live. They might have negative delta G's and be spontaneous, but they still just won't proceed at the rate that we need. And so the enzymes serve a very important role by being those catalysts that accelerate those reactions that allow things to happen on a time scale necessary for life. Now, this is kind of gobbledygook, but enzymes can increase the rates of the biochemical reactions by 10,000 to 10 billion fold. That shows you there's a lot of increase. Examples of this high rate here is going to be like carbonic anhydrase that we talked about in the buffer lecture. It's also superoxide dismutase, which destroys superoxide, and catalase, which breaks down hydrogen peroxide. So we'll see examples of that coming up later. Now, the cool thing about enzymes is that they're very specific. Most chemical catalysts, if you think about the chemical industry, they can have side reactions, they can have non-specific reactions, they can have a number of unwanted processes, and the whole idea of chemical catalysts is to optimize the catalyst and minimize the problems. But enzymes are designed to proceed with very few or no side reactions, very, very few, very nice and specific. And they'll make one product and not other products. There's one reaction they do and there's one product that they make. And so as catalysts, they are very, very efficient. And the other thing about the enzyme catalyzed reactions is the enzyme is designed to carry out or catalyze a chemical reaction and make a certain product. But it's possible for that product to bind to the enzyme and do the reverse reaction and come out as a starting material. Usually chemical catalysts don't do that. So enzymes are really powerful, and there's some enzymes that you want to be reversible, like carbonic anhydrase, where it will use carbon dioxide and make carbonic acid in the tissues, but in the lungs it'll take carbonic acid and make CO2 that can be exhaled. So there's a clinical relevance of enzymes. We'll see this more in the next lecture here, but for pharmacology, when you're working with drugs, drugs will target the active site of enzymes, usually to either kill that enzyme to stop its activity, or sometimes the pharmacology is designed to set up the enzyme to work better. And we'll see coming up. So for example, if you have an aspirin, there's an enzyme called cyclooxygenase. The aspirin will chemically modify the active site and basically kill that enzyme. Sometimes you have an inactive precursor drug that's given and it is in the system, and then an enzyme will take that drug and modify it, and that makes it an active form of the drug. And so you'll see a lot of this in the pharmacology class that starts in block 2. Now you can imagine, because enzymes are so important for these biological reactions, if there's a problem with the enzyme, if it's not present, which is an enzyme deficiency, or if it's not working properly, then you're going to have trouble meeting the needs of the cell. So enzyme deficiency is the cause of many disorders, and we'll talk about that as we go through this class here. Sometimes you can have structural changes in the enzyme that changes the shape, that changes the structure and the function, or it could be you could have an enzyme that's part of a chain reaction, several enzymes in a row, and if one of those enzymes is missing, the whole pathway fails. So we'll be discussing this and seeing how some of the disease states that are enzyme based will give you good examples of that. So this is a really nice way to understand enzymes. So here's an enzyme. I call this the lion, and there's a lion's tooth, and it's got this mouth here or whatever. So the enzyme is a protein, and all of this blob here is just to show you the three-dimensional folded form of the protein. And what's different about this is there's going to be a special place on this protein called the active site. And it's a pocket or a dip or an invagination or something where you have an open area. And that open area is designed to bind a molecule, a starting molecule called a substrate. Old language for enzymes, but the substrate is the molecule that the enzyme will work on. That's the starting material. And the active site here is going to be designed by biomolecular recognition to look like the substrate. And the substrate, when it diffuses, if it goes into this active site, it will recognize the surface of the active site, and there will be an affinity. It will bind because there is recognized complementarity. You'll set up weak interactions. These black lines are showing you hydrogen bonds or hydrophobic interactions or ionic interactions. There is something about the enzyme's active site that will perfectly match that substrate to allow it to bind, okay? Now, here's the thing called a cofactor. We'll talk about cofactors in a couple of lectures. It's the vitamins and the minerals lecture, but the cofactor is going to be a separate organic molecule that has more chemical power than the side chains of the amino acids in the active site. Not every enzyme uses a cofactor, but some enzymes do, and that's why that's there. And the cofactor is important for help positioning the substrate. The cofactor will interact with the substrate and plays a role in the chemical reaction. So what you'll notice now is the substrate finds its way in, it's recognized, and the enzyme will accommodate that substrate to allow the substrate to bind. And you see these weak interactions are set up. Before, this little green patch is showing you the part of the enzyme that attracts the substrate. But when the substrate goes in there, the enzyme says, I will change my shape. I will accommodate your shape so that you can bind, and you can bind with enough affinity that you'll stay here. And so now this is oftentimes called the induced fit. The enzyme is changing itself to allow that substrate to bind properly in there. And the payoff for that is all of these weak interactions. They let off just a little bit of energy, and that accommodates the binding. Well, the enzyme now says, this is not my shape. This is not where I want to be. I want to go back to a more normal shape. And the enzyme will change its shape. It will change its conformation. And as it changes its shape, because there are bonds holding this substrate molecule, and because of the interaction shown here with the cofactor, that causes the substrate molecule to be changed. It gets bent in this case, it gets strained. Now, that's going to be an energy requiring process. It takes energy to modify the substrate molecule. And that's why that's called the transition state. Now, in order to pay that energy cost, over here we have additional weak interactions that will be formed with this transition state complex. Notice here that when the substrate is bound, none of those weak interactions exist yet. Okay, so the substrate has some weak interactions, but there are more weak interactions when you're in the transition state. And energetically, that is favorable and makes it easier for the substrate to go to the transition state. Well, as soon as the substrate here is bent or twisted or stressed or strained or whatever, it can't hold up any longer, and the energy allows that bond here to break, and the circle and the square are now two separate pieces. And the enzyme is designed now to have affinity for the substrate, but lower affinity for the products. So as soon as that substrate goes to products, the enzyme will say, wait a minute, you're not the same molecule that I bound. I don't really have affinity for you, and so you need to leave, and so the products will go. So substrate goes in, enzyme the induced state here, and then you have the transition state, and of course product formation. So as I mentioned before, enzymes have high specificity and selectivity. So here's an enzyme we'll spend a lot of time talking about, and that is hexokinase. Hexokinase is an enzyme in muscle and red blood cells and brain, and its job is to bind a molecule of glucose, and that glucose is going to find its way into the active site, and it's going to be recognized and bound. And when that glucose is bound, there's also a molecule of ATP, which is not shown here, and that ATP will put a phosphate onto the glucose. It will form glucose 6-phosphate. And we're going to see this enzyme a lot, so don't worry about those details. So now you can see the active site, you can see the pocket, and it's hard to tell, but there are residues in here, side chains, that are going to match that glucose molecule very nicely. Look over here. When the glucose molecule is bound in the pocket, because of the three-dimensional geometry of the glucose molecule, the amino acid side chains, these black parts here, are the balls, those little spheres that are right there in the lining. you have aspartates and asparagines and glutamates and stuff. It's all set up to allow a maximum number of weak interactions. In this case, it's hydrogen bonds we're looking at, because we're looking at oxygens and hydrogen and that kind of stuff. And you'll notice that the position is absolutely perfect so that the glucose molecule is positioned in here. Here's the anomeric carbon right there. You know that because there's the oxygen. It's positioned with that anomeric carbon here, facing sort of more towards the outside, and then over here is the glucose 6 position where it's going to get phosphorylated. Okay? Now, what the enzyme says is when the glucose goes in there, the enzyme will recognize the glucose has come in, and it's going to want to form these weak interactions with the glucose molecule. And so this lobe here, this dark gray lobe, will actually fold down and close in and snuggle in around that glucose molecule right there. And of course, you can see the glucose molecule. That's where the ATP will go in and bind and then phosphorylate the glucose. So substrate recognition by these weak interactions, you have the induced fit where the enzyme is changing itself to accommodate that glucose, and then the reaction can proceed. Now we talk about specificity, and specificity just means one specific orientation, as you see here. What about selectivity? Well, here's the carbohydrate galactose, and galactose and glucose are identical except for the configuration of this OH group right here on carbon number 4. For galactose it goes up, for glucose it goes down. Now galactose will go in there and galactose will bind because most of the molecules are the same. The OHs around here are the same. But what you're missing with galactose is with this OH pointing up in the air, it's going to point up over here. And it's going to miss out on all those weak interactions that would be formed with the aspartate 205 and 4 and 231. And so galactose can bind, but it won't form as many weak interactions. It won't have very high affinity, and so galactose is not a very good substrate. There is a galactose enzyme, kinase that will phosphorylate galactose. So there's an enzyme that does that. So there's a couple of factors that can affect enzyme activity, and we'll talk a little bit about that. And the main thing we'll talk about is during the next lecture is going to be the substrate concentration. As a catalyst, the enzyme is going to be present in a small concentration. You don't need a lot of it because it's a catalyst, but you will have very high levels of substrate, and so the substrate will completely swamp the enzyme, or the enzyme will be completely filled up with substrate. And so what you can see from here is, for this blue curve, if you're looking at how fast the enzyme works, the reaction velocity, how fast that enzyme is making product, you'll notice that as the substrate increases, as you move to the right on the x-axis, the substrate concentration gets higher and higher and higher. The enzyme activity gets higher and higher and higher until eventually it saturates. And it saturates because 100% of the enzymes are working with 100% of the substrate, and it can't go any faster. And so substrate concentration is one of the main ways that you can affect the rate of an enzyme catalyzed reaction. We'll talk about allosteric enzymes in a couple more lectures here. They're very similar, it's just they've got one extra physical step that takes place that gives them a difference in their shape here. Now, if you're in a laboratory and you're studying a chemical reaction, one way to change the rate of the chemical reaction is to change the temperature. And we know that the higher the temperature, the faster the reaction will take place. But that's not true for enzymes. Because enzymes are proteins and they have all these weak interactions, we know if the temperature goes up, the vibrational and rotational and translational motion of the atoms changes, and that causes those weak interactions to change, and the enzyme will actually denature, it will fall apart as the temperature gets higher. So our enzymes are designed to operate at body temperature, and you can see you can get a little bit of a change in activity over the normal body temperature range, but really not a lot of change. So temperature is not very viable for us. Now, pH is another way to do this. For most enzymes, of course, they're going to operate at pH 7.4 or 7.2 based on the body. But there's a group of enzymes. These are protease enzymes whose job it is to bind and cut up proteins. They break peptide bonds, and they are activated at different pH levels. And so trypsin is an enzyme, a proteolytic enzyme that operates in the digestive tract. And you know the stomach is acidic, and the acidic environment drops the pH, and that causes the enzyme to be activated. Here's pepsin. Pepsin is an enzyme that gets activated at very low pH in the stomach. Pepsin is the first one that gets activated, and then it will activate trypsin, and trypsin will do chymotrypsin, and so forth and so forth. Now here's an enzyme called alkaline phosphatase. pace, and it's going to be more active under alkaline conditions. Notice the body is at pH 7.4 or 7.2, but if the pH gets a little higher, here the maximum activity is going to be at about pH of 9, alkaline phosphatase. So there are a couple of classes of enzyme. It's fairly easy to remember these if you just think about the types of reactions. And the first kind is oxidoreductases. So that's oxidation reduction reactions. These are going to be enzymes that catalyze reactions where electrons are transferred from one molecule to the next. And we'll spend a tremendous amount of time talking about these reactions in metabolism. So oxidoreductases, it can help the oxidation of a substrate or the reduction, whatever, okay? The next class is called transferases, where you'll transfer a group from one molecule to another or one substrate to another, because we're talking about enzymes. That would be the case of hexokinase, where you transfer a phosphate off of ATP and you put that phosphate onto glucose to make glucose 6-phosphate. That is a transferase, which is true of the class of enzymes called kinases that use ATP to phosphorylate other molecules. The next one is going to be a hydrolase. A hydrolase carries out a hydrolysis reaction. If you have a molecule and you want to add water across a double bond where OH ends up on one of the carbons and H ends up on the other carbon, then a hydrolase is going to do it. Those protease enzymes we just talked about a moment ago are hydrolases. You add water across the peptide bond to break the peptide bond to give you a carboxylic acid and an amine group. It is also true for the phosphatases. As you take a phosphate off of a molecule, you add a hydrogen to one and you add an OH to the other to get a free phosphate in the free molecule. A couple more here. There are lyases, and a lyase is going to carry out the cleavage of a molecule. To lyse something is to split something. So if you had a molecule like ATP, and you split a phosphate off of it, that's like a lyase functionality. So you can see these already, you're seeing that one enzyme, one reaction can be characterized by a couple of different names. We're not going to split those hairs. It's just important to recognize that you can talk about these a couple of ways. Isomerases are going to carry out a rearrangement of a molecule in order to make a new isomeric form. For example, if you consume a D amino acid and you know your body prefers the L amino acid form, there are amino acid isomerases that will change the configuration from D to L so that L amino acid could be used by the body. And then ligases, when you ligate something, you join it together. So a ligase will join these together. Condensation reaction means you bring two molecules and make it one. Usually it's by the elimination of water. It's a very common theme there. So when you make a peptide bond, for example, you are ligating two amino acids, and water is eliminated in the process of making the peptide bond. Okay, so now we need to consider the energy changes in chemical reactions, and we can see how enzymes do what they do. And we need to remind you of the term free energy. And if you remember free energy, that's the quantity of energy that's available in a system to do work. And, you know, this isn't a real rigorous biochemistry class. We're not going to make a big deal of all these energy changes and stuff, but it's important to understand the basic idea and see some fundamental equations that will help you understand that. So the free energy change for a reaction will help determine whether or not a reaction will happen all by itself or not, the spontaneity. If a reaction is spontaneous, that means it will happen by itself without any help. If a reaction is not spontaneous, that means that you're going to have to put energy in. So what is the difference there? The difference is a spontaneous reaction will have one that liberates free energy or gives off free energy, whereas a non-spontaneous reaction, you have to put energy in to make it happen, and that's the difference. Now we talk about this in terms of the Gibbs free energy, or delta G, and this tells you under standard conditions, which is pH of 7 and 1 atmosphere of pressure and body temperature and stuff. And the little apostrophe means it's an experimentally derived parameter here. And that's going to be the energy difference between the products and the substrates. So every molecule will have a certain amount of free energy. The substrate has some free energy, the products have some free energy. And when you transition the substrate to the product by a chemical reaction, you either have to increase the free energy to make the product, or you will get energy out when you make the product. And when you get energy out, when energy is liberated, It's called negative, or the release of energy. So a negative delta G means the reaction is giving off energy, and that allows that reaction to proceed in the forward direction spontaneously. Of course, the opposite is when the delta G is greater than 0, you have to put energy in. The reaction is not spontaneous and will not proceed in the direction shown. Okay. So enzyme catalysts will change the rate at which the equilibrium of the reaction is achieved, but it can't change the position of the equilibrium itself. The catalyst can't change the free energy. That's a fundamental property of these molecules. All the enzyme is doing is changing how the substrate gets to the products and what the energy requirements are to transition between those two states. So here's the best way to show this. It's called an energy coordinate diagram. And the energy coordinate diagram shows energy on the y-axis and the progress of the reaction. So initially you're looking at a substrate molecule, a starting material, and you want to convert that starting material into a product, or the final state down here. And in this case, because the energy is on the y-axis over here, it shows the initial state has a higher free energy. It shows the final state has a lower free energy. And since you define the change in free energy as the final state minus the initial state, you have a smaller number minus a larger number. It's going to be a negative number. So you have a negative, or a liberation of free energy here. And so you would call this reaction spontaneous. So this is going to be a reaction of the peptide bond. This is a protein here, and this is one peptide bond. There's the carbonyl from one amino acid, and there's the amine from the other amino acid. They're joined together by that peptide bond. And you're going to add water across that bond, so you end up with a C double bond with an OH to make the carboxylic acid, and you'll be left with an amine group here, which will be an NH2 and then protonated. But right now you have to break that bond. Well, if you know anything about proteins, you know even though this is a spontaneous negative delta G reaction, our proteins don't automatically fall apart in water. They don't. Something is required to make this reaction happen. And what is required is you have to add strain. You have to bind this molecule, you have to strain it and twist it and get it set up so it's ready to react and have that water come in and bind. And so you have to put energy into the molecule. Remember the line head picture where the induced state and it twisted the substrate? Well, that's what's happening here. The water is going to add across that carbon-nitrogen bond and set up this transition state. Now it's much higher energy here because carbon has 2 oxygens, negative, and you've got this nitrogen here with these 3 bonds, and this is not the preferred state. This molecule would much rather be a carboxylic acid, or would much rather be an amine group over here, or it would much rather be a peptide bond. And so this is not its preferred state. It's energetically higher than normal, but that's the transition state. And when you reach that state, the reaction has to make a decision. The reaction can go back to the initial state and form the peptide and water, or the reaction can go forward and give you the final state. And of course, the energy change is much greater if you go from the transition state to the final state. And since that's the larger energy change, this is going to be statistically the favored direction. And so you're going to see the majority of these molecules are going to go forward in the reaction here. And you're going to end up being the final state by breaking that bond, and that's going to give you the carboxylic acid, and there's the amine group there. Okay, they're not showing I'm deprotonated, but protonated. Now the thing is, initial state, there's the final state. The activation energy is the amount of energy needed to put into that molecule to make it reach the transition state. And then once you go there, you can go down to the final state. So since you started at the initial and ended at the final, the net change is right here. That's the net energy change, or the negative delta G that we've talked about. The activation energy is the cost that you have to pay to make this reaction work. Okay, so again, enzymes, if you think about them increasing the rate of a reaction, how do they do that? Well, there's a couple things that you could do. You could change the temperature here. You know, if the temperature gets higher, you go from blue, which is colder, to red, which is hotter. And of course, as the temperature increases, all the energy goes up. But what you're going to find is the transition state energy is what it is. And so, yeah, you're starting at a higher free energy, and so maybe it's a little bit easier to reach that transition state, but you end up at a higher place here, so there's really no real advantage to doing it by temperature here. And plus, you know, our body can't do that too well because we are in a limited range of temperatures. But instead, what the enzyme says is, I can take that transition state If you remember the line head slide a few minutes ago with all those weak interactions, the substrate will bind and form weak interactions and the enzyme will go through the induced fit. But then as the substrate is moved and twisted and turned, the enzyme active site will make more weak interactions with the transition state than it did with the substrate binding alone. And all of those weak interactions liberate energy. And that energy that is liberated is going to pay the cost of this energy change here. So without the enzyme, in order to reach the transition state, you have to put in this much energy. That's called the uncatalyzed activation energy. But when the enzyme is present and forms all those weak interactions reduce the energy necessary to reach the transition state, and you only have to pay this much energy cost. And as you can imagine, high energy means a slow rate, lower energy means a faster rate. And because of that, it's easier to reach the transition state. You still have a very large negative delta G change here, and that allows you to go forward towards the products here. Okay. So what's very important is down here is notice that the starting energy of the substrate and the final energy of the product is not changed here by the presence of the enzyme. You don't change the free energy of the reaction, so delta G is not affected. And because we know, we'll develop this a little bit later, but we know that the delta G for a reaction is going to be related to the gas constant and the temperature, but here, the natural log of the equilibrium constant. And if the delta G is not changing, well, this gas constant is constant and the temperature is constant, so that must mean that the equilibration constant is not changing either, and that's the case. You don't change the equilibrium, you just simply change the energy needed to reach the transition state, which is going to increase the rate of getting towards products over here. Okay, so when you add an enzyme in on this, it's a little bit complicated because, as you can imagine, there's more going on. Here's the same reaction we saw before. We saw that they're going to add water across this peptide bond, and that's going to take you to the transition state. Okay, so when the substrate binds, the first thing it has to do is find its way. Remember the line slide. The line slide, helps you there. And the substrate has to wiggle its way in, so it takes a little bit of energy to wiggle into that active site there and get the active site to adjust around it. But once you get into that induced state there, the enzyme says, hey, I'm pretty happy because I have made some weak interactions with the substrate. Well, then it's necessary to get that water to add across that peptide bond. And when you do that, you're going to start making these covalent bonds that didn't exist before. But again, the enzyme is going to say, let me make new weak interactions here that didn't exist before. And by making those weak interactions, I'm liberating energy that's going to pay the energy cost of going to the transition state. So you add the OH here, and you're going to add the H, of course. And you're going to reach that final transition state that we see shown above here, where you've got all those weak interactions. And of course, now you've got the decision, do you go backwards, do you go forward? Of course, the favorable is to go in the forward direction here and to break the bond between the carboxyl group and the and the amine group. This is a little bit different because there's a couple steps, and we'll see that in a few minutes here, and that's how it works. So you see there's a couple of steps along here, but overall the same idea is true. So let's introduce these terms that are important, and the first one is going to be binding energy. When the substrate goes in and it finds the active site and it's recognized by that biomolecular recognition, it will create new weak interactions, and those new weak interactions will liberate energy, and that is energy that is free energy that's available to the system. So that is the energy that is liberated upon binding. And there's actually two types here. There is the substrate binding energy, but there's also the binding energy that comes when you form the transition state with these extra weak interactions that are formed. And so it's all of these together that is the binding energy that is paying the cost for the activation energy. The next one is proximity. If you think about this substrate and you have to have some kind of chemical reaction, something has to act on this substrate. It's not just going to break by itself. It's not just going to fall apart. Something has to do work on this molecule. Well, the enzyme, by creating a very favorable reaction site, is going to allow this substrate to come in and come into proximity of all of these side chains that will allow these weak interactions to form. It'll set up proximity to allow this cofactor to interact with the molecule and allow proximity to drive this thing towards the transition state. And the induced fit we talked about previously before, in order to set that up, the enzyme has to change its shape. Okay, it has to change its shape to accommodate the binding of the substrate and to set up these weak interactions. But there's two sides to that. When the enzyme induces its induced fit for the substrate, the enzyme will now go back to its conformation closer to what it had before, and that's going to induce a shape change on the substrate, and that's that strain or the chemical reaction that's taking place. So it works from both perspectives, the enzyme to the substrate and the substrate to the enzyme. And then orientation. What you can see here is when this substrate binds, there's a square and a circle. And the square is designed to bind up at this part and make all of these weak interactions, and the circle is designed to interact with this cofactor here. And so this substrate can bind in any number of ways in the active site, but only if it binds in the correct orientation will the reaction take place. Well, how does the enzyme control for that? Well, because of affinity. When this substrate finds its way into the active site, those parts of the molecule will be attracted to the geometry and the surface of the active site because of those weak interactions. And as the molecule enters in, it's very natural for that square to move up towards the top to make those interactions, which positions the circle to be over here by the cofactor. So binding energy, proximity, induced fit, and orientation are critically important terms. So let's talk about how it works. I love this. I've been doing this for years. So let's talk about the enzyme stickase. And let's say you have a metal stick here, and the cell says, you know what, stick, I need you to break. I need smaller pieces. This is too big. And the stick says, no problem. I can bend and I can break. Well, are you going to have a stick just spontaneously bend? Is it going to bend itself? Is it going to put enough strain in there that's going to allow it to bend so much that it will break? That's never going to happen. The amount of energy it would take to bend that stick enough so that it'll bend and actually break is enormous. Okay, so that's just not going to happen on its own. So let's forget about that. Well, let's create an enzyme, or have an enzyme here called stickase. And stickase is going to be an enzyme that has an active site. So the purple is the protein, and of course the active site is here. And the pocket, or the active site, is going to be lined with little magnets here. These little side chains, the residues of the amino acids, are little magnets. And so when the stick first goes in there, the stick is going to be recognized by the active site because it's going to say, hey, magnets, I'm a metal stick, I like you. And so there will be affinity that will allow this stick to bind. And in fact, you see that right here. Initially, the substrate will go in. It takes a little bit of energy to get it to go in there. The enzyme's got to open up and let it in. But when you form those weak interactions between the magnet and the metal, you get a little bit of energy out. Well, the stick now is in there and it's pretty happy, but the stick says, wait a minute, I see all of these magnets and I'm a metal stick, and I bet if I were to bend, then I would have a whole lot more interactions here and I would actually be in a lower energy state. And the enzyme is saying, yeah, you can bind, and if you bind, I promise I'll give you those interactions and we can go to this transition state. And sure enough, the stick will now bend because it's energetically favorable to do so. You're getting all of these weak interactions forming, which is going to reduce the total energy of the system. And when you reach that transition state now, you're at a much lower energy than you are compared to when there's no enzyme present. And so the probability of reaching the transition state is much greater. And so the probability of making product, of course, is much greater. And because of that, when the stick bends, it's going to have enough strain that the stick will pop and two pieces are formed. And then the enzyme says, you know what? I'm designed to bind a metal rod of this length. I'm not designed to bind these pieces. And the affinity is much lower now, and the metal pieces come out. So that's how that one works. Now, what would happen if the enzyme active site looked exactly like the metal stick? What would happen if they were perfect fits for each other from before? Well, when that metal stick went in, of course, it would take a little bit of energy to get that metal stick in. The enzyme active site would have to open up and let the stick in. But because you're forming all of the weak interactions at the beginning in the straight rigid form here, you would have the energy minimum now. You have all of those weak interactions. And the enzyme and the stick now both have to bend to reach the transition state, so you have more energy. You have to go more than you did before. And so this reaction would be completely impossible. Now, this is really important because you may have an enzyme that works like the state down here, the stickase working the way it's supposed to. But when you design pharmaceuticals to kill enzymes, a lot of times you'll design a pharmaceutical to look just like the active site in the transition state. You want that pharmaceutical to bind and put the enzyme in an energy well down here so that you're stuck in that energy well and the enzyme can't do anything. So try to remember that as we go forward. That's a good way to inhibit enzymes, but that's not at all how the normal enzyme would work. So on the remaining slides, we're going to demonstrate all these different properties using a class of enzymes called proteases, which are enzymes that are designed to cleave peptide bonds, to break that peptide bond. And the one we're going to talk about most is called a serine protease because there is a serine side chain that is the primary agent that does the chemistry here. So the serine proteases are going to be a family of proteases that are going to cleave peptide bonds. And each enzyme in this family is going to hydrolyze a specific peptide or amide bind adjacent to a very specific type of amino acid residues. For example, chymotrypsin is going to cleave next to aromatic side chains, and trypsin is going to cleave next to basic or the positively charged residues. And you'll see most of these are going to show up in digestion. These are released into the digestive tract to break up the protein that we have in our diet. And it's a really beautiful cascade of how these things work. Now, serine proteases are synthesized in the pancreas, which is going to be delivering them to the digestive tract, as proenzymes. These enzymes are very powerful at cleaving proteins, and so you don't want them to be active until they're released into the digestive tract. And so the proteins are initially inactive, but when they get into the digestive tract, they're activated. And we'll see more of that coming up in a little while. So here's the specificity. As I mentioned, there's more members than this, but these three are the best known, best talked about. So here's trypsin, and here's chymotrypsin elastase. And this is not the active site, but rather this is called a binding pocket. And this is going to bind the specific amino acid that's going to help the enzyme know where it's supposed to cut. So trypsin is designed to bind positive and basic residues. There's a negative aspartate that forms those salt bridges here, and when that side chain goes down in the pocket, it is immediately recognized and formed all of these weak interactions. And so this is going to serve to be like an anchor. It's going to anchor that protein substrate that needs to be cut. And it's not shown here, but the peptide bond that needs to be cleaved is going to be right beside that amino acid. So that's the peptide bond that will be broken. Now, chymotrypsin will cut beside aromatic side chains. There's phenylalanine and tyrosine and tryptophan. And so this is a hydrophobic pocket. Very hydrophobic environment. So when the phenylalanine is shown here gets down in there, there's going to be a lot of affinity. It will bind. It's going to serve to be an anchor that will really hold this protein down. And there's the peptide bond that's going to be cleaved. Of course, elastase now is going to be working on small side chains and mostly the nonpolar aliphatic side chains and cut beside those. Same idea here. So these are going to be anchors that hold the substrate peptide in exactly the right position needed for the chemistry to take place. So we can switch over and show you the mechanism here. Now, this is a lot to take in, and it might be difficult, but understand the message and be able to tell the story, and you'll be okay. So this little squiggly stuff here is going to be the enzyme. In this case, we're talking about chymotrypsin, and this squiggly over here is telling you the protein that is chymotrypsin. Now notice here's that binding pocket from the previous slide, and in this case we've got a tyrosine. It's one of the hydrophobic side chains, and so this is a very hydrophobic pocket, and when the tyrosine enters that pocket, it's going to have favorable interactions that help it serve to be an anchor. It will hold that tyrosine in place and allow other weak interactions to start to form with the peptide over here. Now notice there is the peptide bond that's going to be broken. It's shown in those blue-green letters there. And the tyrosine serving as an anchor allows that bond to be positioned exactly correctly, the right orientation with the right specificity. You want to cut this bond and no other, and it's got to be in the exact right orientation to let the chemistry take place. Now, the chemistry is going to take place using the serine. That's why it's a serine protease. And the serine has an oxygen, and oxygens are nucleophilic, and it's going to be attracted to the delta positive of this carbonyl carbon. You can have a nucleophilic attack. Well, The problem with that is some of the electrons on this oxygen are going to be used by that covalent bond with that hydrogen atom. And so it's not really as strong a nucleophile as it can be. Now over here is a histidine residue that just happens to be one bond length away from that hydrogen. And if you remember, histidine is a side chain that can pick up a proton and become protonated. And so this proton now will sense those electrons and will move over, as you see down here. It's now forming a new hydrogen bond interaction that's going to pull that hydrogen away, and that's going to let the oxygen have all the electron density. That electron density will be more powerful, and it will attack that carbonyl carbon, as you see down here. Now, what about this aspartate over here? Well, as soon as that hydrogen moves over, that proton moves over and protonates this nitrogen, that's going to change the electronic configuration of this ring here. And so the aspartate has this negative oxygen that has a hydrogen bond interaction with this hydrogen over here on the ring, and that hydrogen bond will draw that hydrogen off to allow more electron density to participate in this ring and allow this thing to pick up the proton. So by these three residues working together, they're called a catalytic triad. You're making the serine oxygen a better nucleophile by bringing that proton off of that oxygen. So once the nucleophile attacks, you've got a new covalent bond that didn't exist before, and so carbon has its original C double bond O, but it can't have 5 bonds, and so that double bond becomes a single bond by moving those electrons up, and you've got a formal negative charge there. And that formal negative charge is going to be picked up by hydrogen bonds on the surrounding protein. And that's going to stabilize the structure, but those are the weak interactions with the transition state that lower the energy of activation to allow this transition state to be reached more easily. And so now the carbon says, wait a minute, this is not my best configuration. I don't want to be here. It says I could rearrange myself and become much more stable. And so out of all of these groups, it turns out that an amine is the best leaving group. And if you remember the leaving group from organic chemistry, it's going to be which one of these groups is going to be most stable if the bond is cleaved. And it turns out to be the amine. And so the carbon says, I'm going to release those electrons and let the amine have them. I'm going to reestablish this double bond here with the carbonyl carbon, and that's going to set up that structure here. So now you've actually broken the peptide bond. It's now broken. The first product is free to go away. You're left with an amine terminus, but you've got another problem now. You've got a problem where that piece of the protein here with the carbonyl is still attached to that serine. And remember, enzymes are catalysts. They can participate in the reaction, but they're not supposed to be changed by the reaction. So what we need to do now is break this bond. We have to hydrolyze the enzyme carbonyl bond. You'll notice that the proton that used to be on the histidine side chain went over here to be with the amine terminus over there. Well, there are water molecules. This is a water environment, and if a water molecule comes in proximity, the same thing can happen that we saw with the serine. The histidine can pull a proton off of that, and it can leave all the electron density on that oxygen, and now that oxygen is in the perfect orientation, it's the perfect geometry, it's close together, and then it can have a nucleophilic attack on that carbon atom right there, and it will form another bond, and that C double bond O can become a single bond like we saw before. It's the same thing, it's just using water instead of the serine. And again, you have a situation where carbon now has these 4 bonds, but now it has 3 oxygens. And carbon doesn't like to have 3 oxygens. And so it's going to rearrange itself structurally to accommodate that. And it says, what is the best leaving group? And of course, the best leaving group would be a carboxylic acid. So to do that, it says, I will break the bond between this carbon and the serine oxygen, and that will leave me with the OH of my carboxylic acid, and that will give me the double bonded oxygen for my carboxylic acid. So these are the rearrangements that we see, and you're left with the carboxylic acid. You're left with the serine now regenerated in its original form. The proton that was on the histidine side chain now goes over to be a part of that, the serine over here, okay? So again, proximity, orientation, active site, transition states, all of it. Okay, so there's another class of proteases, and you'll see this more in immunology because you see these with virus particles, but these are aspartyl proteases. And the aspartyl protease is very similar. You have a peptide bond on a protein that you want to break. So here the protein that is the substrate is shown in red. And this is the peptide bond that you are going to break. Now, you have aspartate residues that are here. And if you remember, aspartate residues have that carboxylic acid. And so they can pull a proton off of water, and that can activate the oxygen to be a nucleophile. And when the nucleophile attacks, you can have the addition of a water onto that carbon. You get a structure that's very similar to what we saw for the serine proteases. You've got 4 bonds, but you've got 2 OHs. And carbon does not want to have 2 OHs on it. And so you can have a structural rearrangement where the peptide bond will break, and the hydrogen, the proton will go over here back to that aspartate like before, and you're going to donate a proton off of the other aspartate to give the amine group what it needs, and you've broken that peptide bond there. So instead of using a serine, you're using aspartates, but you're activating water as a nucleophile, which is very similar to what we saw. Okay, here's a couple of questions, and we'll still go through these now because it's early in the lecture series, but I'd like to do a little bit less and have you do more of these as an active exercise. So the first question is lactate dehydrogenase, an enzyme we'll talk plenty about in metabolism. Its job is to take 2 electrons and 1 proton away from the molecule lactate to form the molecule pyruvate. This happens in red blood cells and in liver, and it's a really important reaction to interchange lactate and pyruvate. But here, lactate is giving away 2 electrons and 1 proton to form pyruvate. What type of enzyme is lactate dehydrogenase? So to answer this question, you have to ask yourself, what do I remember about electrons changing place, and what kind of an enzyme is that? Or if you can't remember that, you can come down to the answers and say, well, what is a hydrolase? Oh, that does the water, and isomerase changes the structure. Ligase is going to bring 2 things together. Oxidoreductase is going to change the oxidation. Oh, there it is. Okay, so that's how you want to go about these 2 different forms. This is a little bit simpler than a test question, but it's a good way to test a basic definition. This is a really good one. This is actually very consistent with the level of knowledge needed for USMLE. Here's a patient that's being treated for a mutation in our hexokinase enzyme. We talked about hexokinase a lot. That's the one that binds glucose and ATP and has a phosphorylated glucose product. Now, instead of a glutamate residue at position 256, she has a threonine residue. So this is asking you to remember a little bit about glutamate. That's one of those ionizable side chains. It's got a carboxylic acid, and it's going to be deprotonated. It's going to have a negative charge. And then threonine, you got to remember a little bit about that. It's one of the polar but uncharged amino acids. So it doesn't have a negative charge, and it's not a carboxylic acid. So automatically you see there's a pretty big difference there. Which of the following would be a likely effect on glucose binding to this altered enzyme? Well, in this case, you don't really have to memorize the structure or memorize the amino acids to recognize that when a substrate goes into an active site, it's going to be recognized by biomolecular recognition. The side chains will match the structural features of the substrate. And so if you change those amino acids that are there, especially one that is very polar and has a formal charge with one that's less polar and has a very different shape, well, you're not going to have the same geometry, you're not going to have the same fit, and so you would expect the affinity for the substrate to be a bit lower. So what you need to do is find an answer down here that has something to do with affinity change. Glucose won't bind at all to the enzyme. Well, that's not true, right? Because there's lots of residues that make the active site. And so you're just changing one of them. Glucose will bind, but with a lower affinity than normal. That sounds pretty good. Let's put a pin in that one. Glucose will bind, but with higher affinity. It's hard to see how an amino acid change like that can make something have a higher affinity. Glucose will act as an irreversible inhibitor. Well, no, it won't, because it's the substrate. Now, to make it easier, if there's an exam question like this, of course, what you'd want to have is some information about, a picture. I like to show pictures like this, and you'll see. So we're asking about glutamate residue at 256, and 256 is right here, that one. And you can see how important that residue is because it makes 3 hydrogen bonds. And if you lose that carboxylic acid with a negative charge, you're going to lose 3 of those hydrogen bonds, which is really going to affect the affinity. You see threonine, this is the side chain right there. It doesn't even look like glutamate. Can't do it. And here's one last one. Here's a patient that has a defect in her chymotrypsin enzyme. In place of the catalytic triad histidine residue, she has a phenylalanine residue. How does this difference affect the function of her chymotrypsin enzyme? So again, even if you don't remember anything about the structure or the chymotrypsin mechanism or the enzyme active site, you just remember something about these amino acids. And if you remember the catalytic triad, the serine residue is the nucleophile, and the histidine is able to activate that serine because the histidine can pick up a proton from the serine side chain. If you recall, phenylalanine is a nonpolar. It's an aromatic side chain, and it is not ionizable, so it cannot pick up a proton from the serine residue. And because of that, the serine residue will not be activated, and so the serine residue will not be a very good nucleophile, and so this catalytic triad will not work very well. So you're going to try to find an answer that has something to do with a loss of function or something along those lines. And I think I see one. And in fact, when you see the detail, it's a little bit easier. Here's the catalytic triad. There is the histidine residue that takes that proton away from the oxygen to let that serine oxygen be a stronger nucleophile. Here is phenylalanine. It's non-ionizable and it can't pick up that proton. So you have a loss of function. Thank you.